Hello dear students, this is Dr. Adwet and welcome to lecture number 5 of Cell, the Unit of Life. As always, let's have a quick recap of what we have done so far in the previous 4 lectures. In lecture number 1, we spoke about why are there so many Latin words in our subject. We spoke of words like bios, which means life, logos, which means study of, auto, which means self, hetero, which means different, trophos, which means to eat, botan, which means plant, zoon, which means animals. We saw definitions like biology, which means the study of life, botany, the study of plants, zoology, the study of animals, and we also saw the most important definition, which is the definition of cell itself. Cell is the structural and functional unit of life. Cell is the smallest particle which is going to show the property of life. Hence, cell is called as the structural and functional unit of life. In lecture number 2, we started off with more Latin words like micro, which means very small, macro, which means small, mega, which means big, magnum, which means very big, scopion, which means to see, and you have cyton, which means cell. Then we saw what is the history of microscopy. We saw how microscopes evolved, who was Robert Hooke, who was Anton von Leeuwenhoek. We then went on to discuss what is cytology. Cytology is defined as the microscopic study of cell, and it deals with all the cell organelles, how they are going to multiply, how they are going to be structured, how is the cell going to divide. All of that comes under something called as cytology. We also saw in lecture number 2 what is the cell theory. Cell theory as developed by Schleiden, Schwann and Virchow, which says that number 1, all living things are made up of cells or products of cells, and number 2, that new cells come from pre-existing cells. So that was lecture number 2. In lecture number 3, we started off with these things, or we discussed these things which you see on your screen right now. So we have what is the concept of metabolism. If you take small things and make something big out of it, it is called anabolism. If you take something big and break it into smaller pieces, it is going to be called as catabolism. And these are the only 2 main types of reactions happening inside a living organism. So both of them together, jodna matlab anabolism, todna matlab catabolism, both of them together are called as metabolism. We then saw what are the parts of a cell. That cell is living material which is called as protoplasm. It has a boundary called as a protoplasmic membrane or a cell membrane or a plasma membrane, inside which you are going to have the cytoplasm and the nucleus. In the case of a few cells like plant cells, they are protected by a non-living tough covering which is going to be called as a cell wall. Cell wall and cell membrane are 2 very different things. Cell wall is dead and cell membrane is living. Then we saw the 2 Hindi anecdotes which explain to you about what is the main core concept of genetics. And here I quickly once again want to revise that thing, that there are going to be cupboards which are going to have books, which are going to have pages जिसमें किसके बारे में लिखा है और क्या लिखा है. So you are going to have a nucleus which is going to have chromosome which is made up of DNA which comprises of genes which is going to have the alleles. This is something that I will keep repeating even when we do genetics, so do not forget this part. And then we saw what are the characters of a cell. So what are the characters or characteristics of a cell? A cell is the structural and functional unit of life. It is made up of living material called as protoplasm surrounded by a protoplasmic membrane. It comprises of a controlling center which is called as the nucleus and an executive center which is called as the cytoplasm. The nucleus contains the genetic material in the form of DNA and RNA that is responsible for transmission of hereditary characters. The cytoplasm contains the various cell organelles which carry out the metabolic activity. The cell is capable of growth, it is capable of division, and if need be, the cell can also repair itself. So these were the characteristics of a cell that we saw. And we ended lecture number 3 with what is the concept of a eukaryotic cell and what is a prokaryotic cell. So plant cell and animal cell are eukaryotic cell, whereas bacterial cells are going to be prokaryotic cell with a primitive nucleus. The eukaryotic cells, plant cell and animal cell, have a true organized nucleus. So that is what we saw about prokaryotic and eukaryotic cell. In lecture number 4, which is the last lecture, I told you that currently for the next few lectures we are going to speak only about the eukaryotic cell. We are not going to discuss prokaryotic cell. And as you can see on your screen right here, the eukaryotic cell is going to have a plasma membrane, cytoplasm and nucleus. In the last lecture, we did everything related to the nucleus. So let's quickly revise everything related to the nucleus. So the nucleus was first observed in 1831 in orchid leaves, especially the epidermis of the orchid leaves, by this person called as Robert Brown. In somewhere around 1882, you have this other person called as Walter Fleming, who saw that the central part of the cell can be stained by basic dyes, and that central material which he was able to see in the microscope, he termed it as chromatin. I had explained to you last time that chromatin is nothing but DNA plus some proteins. So DNA plus some proteins together can be called as chromatin, and this chromatin, which is basically the material of the nucleus that is stained by the basic dyes. So it was coined by Walter Fleming. Walter Fleming is also the person who coined the term mitosis. Moving on, we saw that this cell is an animal cell, and the reason why we are calling it as an animal cell is because it has clearly got no kind of cell wall, as you can see. So that was the main clue as of now, which was telling you that this was an animal cell. We can also see that the animal cell has a nucleus and has a lot of these cell organelles which are going to be present in the cytoplasm. If I was to take the animal cell and highlight just the nucleus and magnify it, this is how it appears to be. And in this case, you can clearly see that there is one single nucleus in this cell, but in many cells in nature, there may be variations to this. For example, RBCs have no nucleus at all, whereas there are going to be many cells, like some cells in the liver, cells in the muscle, which are going to have multiple nuclei. But the norm is one cell, one nucleus. Now, the nucleus has a nuclear membrane, and this nuclear membrane helps to keep the nucleoplasm separate or compartmentalized from the cytoplasm. So, न्यूक्लियस का मटेरियल अंदर रहेगा, साइटोप्लाज्म का मटेरियल बाहर रहेगा, बीच का जो बाउंड्री है विल बी कॉल्ड एज द न्यूक्लियर मेम्ब्रेन. Inside it, the nucleoplasm can also be called as the nuclear matrix. So there are two things making up the nucleus: the nuclear envelope, and within it you are going to have the nucleoplasm, also called as a nuclear matrix. If we were to take a section of the nuclear membrane and magnify it, we can study it in more detail. In here you have the nuclear matrix, and here you can clearly see that there are openings on the nuclear envelope. There are openings through which material can go inside and outside, and those openings are called as the nuclear pores. Here you can also clearly see that the nuclear envelope comprises of a outer nuclear membrane and an inner nuclear membrane with the perinuclear space in between. This space is about 10 to 50 nanometers. We also saw that there was a line in NCERT which stated that at a number of places the nuclear envelope is interrupted by minute pores which are formed by the fusion of its two membranes. To explain this to you, I had shown you this diagram. So I hope you remember that at places where you have the nuclear pores, the two membranes are going to join. And the newer view of this is that right here you are also going to have a lot of other proteins called as the pore complex or the pore proteins. So that is what you see in the previous picture. Now, how does the actual electron microscope images look like? This is the nucleus, and here you can clearly see on its surface these little things here that you see are nothing but the nuclear pores, which on further magnification appear like this. So that finishes everything that you needed to know about the nuclear membrane. Talking about the nuclear matrix, the first thing that the nuclear matrix contains is going to be the chromatin. So chromatin is the material of the nucleus that can be stained by basic dyes, and here I explained to you something about DNA. So we discussed that the DNA is a double helix or a helical structure, which is pretty much the same structure in the case of a eukaryotic cell or a prokaryotic cell. But the difference is that in the eukaryotic cell, it is called linear because you can find the ends of the DNA. In the case of prokaryotic cell, you cannot find the end. It appears like a big circle, and that is why it is going to be called as circular DNA. So eukaryotes are going to have linear DNA and prokaryotes are going to have circular DNA. Then we saw that chromatin was one component of the nuclear matrix. The other component was something called as the nucleolus. The nucleolus was a spherical body found inside the nucleus. It does not have any boundary, so the material of the nucleolus is continuous with the nucleoplasm. The nucleolus is something which helps in protein synthesis. In the case of a cell that is actively making a lot of proteins, there may be even more nucleoli in the nucleoplasm. So that is what you need to know about nucleolus. So a eukaryotic nucleus is going to be a true organized nucleus with the presence of a nuclear membrane. It is going to be differentiated from the cytoplasm. So nucleoplasm is differentiated from cytoplasm. It will always contain more than one chromosome. It has linear DNA. Nucleolus will be present, and it is the controlling center of the cell and responsible for transmission of hereditary characters. So here we also discussed about the differences between a eukaryotic nuclei and a prokaryotic nuclei. So a true organized nucleus is present in eukaryotes, a true organized nucleus is absent in prokaryotes. In the case of prokaryotes, it is called as a nucleoid. We saw that here the nuclear membrane is present, here the nuclear membrane is absent. The nucleoplasm is differentiated from cytoplasm, nucleoplasm is not differentiated from cytoplasm. It is continuous. Here you are going to have always more than one chromosome, here you will only have one chromosome. Nucleolus is going to be present, nucleolus is going to be absent. Here you are going to have linear DNA, here you are going to have circular DNA. So these were the differences between a eukaryotic nuclei and a prokaryotic nuclei. Then we saw what does the NCERT speak about the nucleus, and we discussed all the points. I am once again quickly going to read through them for your benefit. Nucleus as a cell organelle was first discovered by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. That was around 1882. Normally, there is only one nucleus per cell. Variations in the number of nuclei are also frequently observed. The interphase nucleus of a cell when it is not dividing, has a nuclear envelope and it has a nuclear matrix or nucleoplasm. The nuclear matrix of the nucleoplasm has chromatin and has nucleoli. Chromatin are highly extended and elaborate nucleoprotein fibers, basically DNA and protein, and nucleoli are one or more spherical bodies which may also be found in the matrix. As far as electron microscopy is concerned, has revealed that the nuclear envelope consists of two parallel membranes with a space of about 10 to 50 nanometers in between, called as the perinuclear space. It forms a barrier between the materials present inside the nucleus and that of the cytoplasm. At a number of places, the nuclear envelope is interrupted by minute pores which are formed by the fusion of its two membranes. And these nuclear pores are the passages through which material can go from inside to outside and from outside to inside. The next thing, the final thing about the nucleus is the nuclear matrix, which, as you now know, is going to comprise of chromatin and is going to comprise of the nucleolus. So first, the nuclear matrix or the nucleoplasm contains nucleolus and chromatin. Chromatin, a highly extended and elaborate nucleoprotein fibers, and nucleoli, which are spherical structures in the nucleoplasm. The content of the nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane bound structure. Nucleus is a membrane bound structure, nucleolus is not a membrane bound structure. It helps in protein synthesis, and larger and more numerous nucleoli are present in the cells actively carrying out protein synthesis. So in lecture number 4, as far as protoplasm is concerned, there were three things. There was a nucleus, there was the cytoplasm, and there was the plasma membrane. In the last lecture, we finished everything related to the nucleus, and now it's time to do the cell organelles. So, dear students, today's lecture starts now. I hope everybody is clear with the previous 4 lectures that we have done. Majority of you have already been doing the assignments properly. All of you have been sending me the assignments. All of you seem to be on the right track and are taking the necessary effort required to understand whatever we have done so far. So I'm very happy with everybody's progress. Everybody seems to be excited and seems to be working hard. Great. So let's start today's lecture. Now, today's lecture begins with a list of more Latin words. So the first words that we have to discuss today are inter or meso, intra or endo, exo, epi or peri. So the word inter or meso means between. So if there is something present between two things, it can be called as inter. For example, if you are going to have a cell and another cell, this space here in between the two is going to be called inter, or to be more accurate, it can be called as intercellular space. So the word inter or meso basically means beech ka, in between. If you were to take this cell again, whatever material is present inside can be called as intra or it can be called as endo. So inter or meso means beech ka, intra ya endo means inside, within. So inter is in between, intra is within. Do not get these two confused. An easy way to remember that is inter-college competition or inter-school competition is very clear that it is in between the colleges or in between the schools. So easy to remember, don't get confused. Inter means beech ka and intra means andar ka. Intra can also be called as endo and inter can also be called meso. Now, if intra or endo is inside, then the words extra, exo, epi or peri means outside. So I said 4 words. The words extra, exo, epi or peri. These 4 words mean outside. They have slightly different meanings, the nuances which I will explain now, but all of them mean outside. Now, extra or exo simply means outside. The word epi usually means outside and above. Outside and above. So as you can see in the cell here, if you see above it, I have written the word epi. So the word epi means outside, but it means outside and upar. And lastly, you are going to have the word peri, which also means outside, but it means outside and surrounding. It means outside and surrounding. So the word exo, epi, peri, extra, all of them mean outside. The word extra or exo simply means anywhere on the outside. The word epi means outside but above something, and the word peri means outside and surrounding something. So I hope that these 3 words are now clear to everybody. Inter or meso means in between. Intra or endo is going to mean inside or within. Exo, epi, peri is extra, exo, epi, peri is going to be outside, where exo or extra simply means outside, whereas epi means outside and above and peri means outside and surrounding. So are these 3 words clear with everybody? Okay, let's move ahead. Now the next word is phyla. Now the word phyla is Latin for leaf. So there are other words also for leaf, but as of right now, let's just remember phyla. So phyla is the Latin term for leaf. You are going to have plastos. The Latin word plastos means formed or molded. Formed or molded meaning कि अगर आपने कुछ मेहनत से बनाया है, जिसको आपने बराबर से कोई खांचे में ढाला है, if you have formed something properly or molded something, the Latin word or the ancient Greek word for that is plastos, made carefully with a proper structure in mind. The next word is amylos. Amylos is the Latin word for carbohydrates or starch. Mostly the starchy vegetables that you know of, like you are going to have potatoes. So amylo basically refers to starch, carbohydrates. Then you have elluro. Elluro is the ancient Greek word for wheat flour, गेहूं का आटा. So गेहूं का आटा, which is rich in proteins, the Latin or the ancient Greeks had a word for it which was elluro. And then the Greeks also had a word for olive oil, which was elion. So I don't know how many of you know this. In Greek cuisine, they use a lot of olive oil because olives are commonly grown in that Mediterranean region. So their word in their language, in their ancient Greek language for olive oil is basically elion. So amylos is starch, elluro is protein, and elion is going to be olive oil or oil. The next word is thulakos, and thulakos is the Greek word for pouch, as you can see here. So any pouch like structure like this is going to be called as thulakos. Now the interesting thing here is that all pouches will have a cavity inside through which you are going to put something in that pouch. That cavity in English is going to be called as lumen. And lastly, you are going to have lamella or lamina, which simply means a sheet. A thin sheet, like a sheet of paper, a thin sheet can be called as lamina or it can be called as lamella. So we've seen now a long list of these Greek and Latin words. Let's quickly revise them because all of these are going to be used in today's lecture. Inter or meso means in between, intra or endo means inside, exo, epi, peri means outside, exo and extra means outside, epi means outside and above, peri means outside and surrounding. Phyla means leaf, plastos basically means something which has been formed or molded with care. You are going to have amylose, which means starch or carbohydrates, eluro, which literally translates into wheat flour or with reference to proteins. You are going to have eleon, which is oil or fats. You have thulakos, which means pouch like structures. The cavity or the space inside anything here can be called as a lumen. And finally, you have thin membranes or sheets which are called lamella or lamina. So are all these words clear to you? Excellent. The next list of Greek words, so still not over, we still have a few more words to do. So colors in Latin. The Latin word for color itself is chroma. So chroma means color. And as far as colors are concerned in Latin, we are going to discuss the 5 main colors and they are Latin names. You have erythros, which is red. You have leukos with a K, which is white. So please notice here you have leukos and there is a K here, which means white. You are going to have lutos with a T. So this is not the same word, it's a different word with T. And lutos is yellow. So you have erythro, which means red. You have leukos, which means white. You have lutos, which means yellow. And then the ones which you probably know, chloros is green and you are going to have cyanos, which is blue. I think immediately now your brain must be able to put 2 and 2 together, that chloro is green and phyla means leaf. So green leaf are green because they must be having something called chlorophyll. Clear? So which are the names of the colors in Greek or Latin? Chroma means color, erythro means red, leukos with a K means white, lutos with a T means yellow. You will have chloros, which is green, and you have cyanos, which is blue. So if this all Latin words are clear, now let's start talking about pigments. So what are pigments? Pigments are basically chemicals with color. So any chemical which has color, perceivable color, will be called as a pigment. Plants have a lot of pigments because clearly plants have color. So now I'm going to show you an aerial picture of a beautiful forest. And as you can see in this forest, right in the center, you also have this lake. Have a look at all these trees surrounding the forest. You will notice that all these trees have a lot of colors as far as their leaves are concerned. To show you another image, have a look at this one. So you can clearly see that all of them have leaves and they have dark green, they have light green, they have yellows, they have reds. So if I was to see this same thing from the top view, the same forest right looking down from top, you can clearly see all the colors that we are speaking about. Dark green, light green, yellowish, you are going to have reddish, dark red. And on this forest floor, if you were to literally walk on this forest, you would see that there are a lot of leaves lying down, and if you were to just collect them, you will see that there is so much variety in the color. So leaves are not just green, they are going to have a lot of chemicals with color, and these chemicals with colors are called as pigments. What are these chemicals with color called? Pigments. So if we were to take a few of these leaves and arrange them, you can see that you go literally from dark green to around red, and this is now a completely dead leaf which has almost decomposed, which is brown. So you can see the whole spectrum of colors that leaves might have. That means that these leaves are going to have various chemicals which are giving them this color. So now let's talk a little bit about these chemicals with color which are called pigments. So if we were to take these leaves, you will see that these leaves have green tones, or the color, the predominant color is green. Whereas here you can see these leaves which are transitioning between yellow, orange and red. So the two main chemicals with colors, pigments in the leaves are chlorophyll and carotenoids. Chlorophyll is basically the green pigment and carotenoids are the pigments which are yellow, orange or red. As far as chlorophyll is concerned, chlorophyll is a fat soluble chemical. So it's a chemical with color. Technically it is called as porphyrins. So in chemistry it is a group of chemicals called porphyrins. We will discuss more about this in a future chapter. As of right now, just remember that chlorophyll are basically compounds which are called as porphyrins and they are fat soluble, and of course as you now know they are green in color, chloros meaning green. The carotenoids are also fat soluble and chemically speaking, as far as their chemistry is concerned, they are tetraterpenes. They are tetraterpenes. Now right now it is beyond the scope to explain to you that what is a porphyrin or a pyrrole ring and what is going to be tetraterpenes. So at the moment just remember these names. Once you build your knowledge a little bit more, these things will become easier. So as of now just remember that chlorophyll are fat soluble porphyrins and carotenoids are fat soluble tetraterpenes. They, as I just said, are yellow, orange and red. In fact, carotenoids are called carotenoids because of the orange color of the carrot. So the pigment which gives carrot the orange color was named as carotenoids, and then they found that carotenoids can range, these tetraterpenes can range from yellow, orange to red. Carotenoids are basically of two main types. There is going to be something called as carotene and there is going to be something called as xanthophyll. Now the main difference between carotene and xanthophyll is the presence or absence of the oxygen molecule. Carotene does not have an oxygen molecule, it's a pure hydrocarbon. So if I was to tell you the formula for carotene, it is C40H56. So C40H56 is carotene, whereas xanthophyll has oxygen. So it is the same carotene but with oxygen. So it's C40H56O2, that is xanthophyll. Carotene is more towards orange, whereas xanthophyll is more towards yellow. So this chart discusses pigments, chemical compounds with color that are found in plants. Mostly there are two types, as you can see right now. They are going to be chlorophyll and they are going to be carotenoids. Both of them are going to be fat soluble. Chemistry, as far as the chemistry is concerned, these are tetraterpenes and these are porphyrins. Here you have yellow, orange and red color, whereas chlorophyll is usually green. The two main carotenoids are carotene and xanthophyll, with the difference being that this has no oxygen and this has oxygen. Carotene is more orange and xanthophyll is more yellow. So are these pigments clear with everybody? So what have we discussed so far? New things that we've discussed in today's lecture. We have seen a list of Latin words, Greek words. We have discussed what is the Greek and Latin name for colors, and we now saw what are the two main types of pigments, colors or chemicals with colors, which are going to be found in the case of a plant. So that, dear students, brings us to the cell organelles of a eukaryotic cell. So there are a lot of cell organelles and you may even have heard about them when you were in school, like plastids, mitochondria, ribosomes, endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, cytoskeleton. We are going to be discussing each one of these in detail. So as of today, let's start with the first one, which is going to be the plastids. So now, what does NCERT say about plastids? Now before we start off with what NCERT says about plastids, it should be clear that plastids are called plastids because they were thought to be specially formed or molded within a plant cell. So they were given the name as plastids, and these plastids inside the plant cell on the basis of their pigments. So plastids are basically cell organelles which are going to have the pigments. Based on their pigments, they can be of three main types. You have chloroplast, chromoplast and leucoplast. Chloro, chromo, leuco. I think this should already give you a hint about why they are so called. So chloroplast are those plastids which will have the green color. Chromoplast will be those plastids which will have the colored pigments, chroma meaning color, and leucoplasts are going to be the ones which do not have any pigments. So they are colorless or they are white, leucoplast. So chloroplast are those plastids which contain mostly chlorophyll and some amount of carotenoids, and this is how they will basically look, mostly green. Chromoplasts have mostly carotenoids and they are going to appear reddish, yellowish, orangish. You will have leucoplasts which have no pigments and are colorless. So plastids, special organelles which are formed inside a plant cell based on their pigments, can be classified as chloroplast, chromoplast and leucoplast. Green with color and white with no color. Now the last group, leucoplast, is further classified into three. So leucoplasts do not have pigments. So what do leucoplasts do? They will store something for the plant. So leucoplast, instead of storing pigments, will store something else for the plant, and they are of three types. The leucoplasts are of three types. You have amyloplast, ileoplast and eleuroplast. Can you now just take 10 seconds to think why they are so called? Amyloplast, amyloplast, ileoplast, eleuroplast. Can you just take 5 seconds to think that? Right. If all of you have been paying attention for the last 15 minutes, you now know what these three leucoplasts, are going to be storing. Amyloplast will store starch, so mostly in the case of potatoes. You are going to have ileoplast which will store, that's right, it will store oil. And lastly, you are going to have eleuroplast which is going to store proteins. So this, dear children, is the first cell organelle that we are discussing in a eukaryotic cell, in a eukaryotic plant cell, which is going to be plastids. Plastids are going to be of three types: chloroplast, chromoplast and leucoplast. Leucoplast will have no pigments, they will store material for the plant cell. They are going to be amyloplast, ileoplast and eleuroplast. So I hope this chart now is extremely clear to everybody, and out of this we have to discuss chloroplast in detail. So majority of the chloroplast of the green plants are found in the mesophyll of the leaves. So as you can see from this symbol here, this line is directly from NCERT, that in the case of green plants, majority of the chloroplast are found in the mesophyll. Now again, take a few seconds to think about the word mesophyll. What is the meaning of the word meso? In between. What is the meaning of the word phyla? Leaf. So mesophyll basically means in between in the leaf, or in between the leaf. So if you were to take a leaf, here you can see we have taken a leaf and we have cut it. In the cut section of the leaf, you will see that this is the upper layer. This is the upper layer, can be called as the outer epidermis. This is the lower epidermis. So whatever you see here, in between the upper layer of the leaf and the lower layer of the leaf, whatever you see here are called as mesophyll cells, and inside each of these cells, whatever green particles you are seeing here, whatever green particles you see here, they are basically the chloroplast. So NCERT says that majority of the chloroplast of the green plants are found in the mesophyll cells of the leaves. In fact, 20 to 40 chloroplasts per cell in the mesophyll. So every cell here every cell that you see here, every cell that you see here can have anywhere from 20 to 40 chloroplasts. 20 to 40 chloroplasts. There are other cells like the Chlamydomonas, which is a green algae, and this is how it looks under the microscope. This is an illustration of Chlamydomonas, and you can see that they only have this one chloroplast. Can you see this here? In the diagram you can see it more clearly. This thing which you see here is the chloroplast. So Chlamydomonas, which is a green algae, is an example of one chloroplast per cell. So just like how nuclei can vary in number from cell to cell, even chloroplast can vary in number from one in a cell like this one, which is going to be Chlamydomonas, to the mesophyll cells of most green leaves, which are going to have anywhere from 20 to 40 chloroplasts per cell. The next thing is that chloroplast can be lens shaped, oval, spherical, discoid, or ribbon like organelles. So they can be lens shaped like a lens, like a convex lens. They can be oval, they can be spherical, they can be oval, they can also be like a disc, they can be discoid, they can be like a disc, and lastly they can be ribbon like organelles. I'm sure everybody must have heard about Spirogyra. So this here is Spirogyra. This is one cell, this is another cell, this is another cell. So just take a look at this one single cell and now follow my mouse. Starting from here, this entire thing, as I'm going through here, this entire thing is one single chloroplast, and this one single chloroplast is like a ribbon. So chloroplast can be lens shaped, they can be oval, they can be spherical, they can be discoid, or they can also be ribbon like. So we saw that chloroplast can have different shapes. We saw that chloroplast can also have different number. Now let's have a look at a typical single chloroplast. So we see here is a plant cell, and now I think that it should be clearly obvious why this is a plant cell. It is going to have the cell wall, as you can see, and these organelles here, these green colored organelles that you see here are the chloroplast. So now once again let us take the plant cell, and these highlighted areas which you see here are the chloroplast, and let me magnify one of them for you. So if we were to take a typical chloroplast, the size is going to be about 5 to 10 microns in length and about 2 to 4 microns in height or width. So it's about 5 to 10 microns and about 2 to 4 microns. Of course, it will vary greatly in size from chloroplasts of different plants and different cells. So there is a great variety here even in the size. So remember that chloroplasts can vary from 1 in a cell to up to 40 in a cell. As far as the chloroplast is concerned, it is also going to have a variable size. It can be anywhere from 5 to 10 microns in length to about 2 to 4 microns in width. As is clearly obvious from this diagram, there is a outer membrane and there is a inner membrane. So chloroplast is a membrane bound organelle just like the nucleus. Please remember that you are going to have nucleus which is membrane bound, nucleolus which was not membrane bound, and now we are doing chloroplast which is found only in the case of a plant cell which is also going to be membrane bound. Here also there are going to be two membranes, and the inner membrane is less permeable. Now NCERT just mentions this point and gives no detailed information about why it is less permeable, etc., etc. So all of that is going to be discussed in another chapter in 11th standard called photosynthesis. So just like you have the chapter of cell that we are doing right now, there is another chapter called as photosynthesis, which is going to take about 10 to 12 hours to teach. So when we do that topic, you will understand why it is less permeable. But here NCERT just mentions that it is less permeable, and that's about it. So for the time being, we will just remember that it's less permeable. Why so? We will learn in the chapter of photosynthesis. So there is an outer membrane and an inner membrane which is less permeable, and the space in between the two will be called as the intermembrane space. So now it should be clear why it is called as the intermembrane space. So this area here, here, all of this area including this area here is going to be called as the intermembrane space. Next, whatever is inside the inner membrane. So whatever you see here, all of this. So ये बाहर का membrane है, ये बाहर का membrane है, ये अंदर का membrane है, और ये जो कुछ भी तुमको यहाँ पे दिख रहा है, whatever you see here, which is inside the inner membrane, is going to be called as the stroma. Is going to be called as the stroma. So the space inside the inner membrane is called as the stroma. So a chloroplast is about 5 to 10 microns in length. It is about 2 to 4 microns in width. It will have an outer membrane and an inner membrane. The space between the outer and inner membrane is called intermembrane space. And inner membrane ke andar you will have all the material which is called as the stroma. So like nucleus ke andar you had the nuclear matrix. Inside the chloroplast you have the matrix of the chloroplast which is called as the stroma. The stroma contains the various chemicals required for synthesis of carbohydrates and proteins. So in the stroma here, you already know right from the first lecture that plants do photosynthesis, meaning they make carbohydrates. So in a plant cell, where do you make carbohydrates? In the chloroplast. So chloroplast basically is green in color, takes light energy and makes glucose. So where does it do that? Here, inside the chloroplast. So what is the inside of the chloroplast called? It is going to be called as stroma. So stroma contains chemicals required for synthesis of carbohydrates and proteins. Clear? Okay, moving on. You will also notice that here there are these little tubes which are connecting a few things inside the stroma. Now these little tubes which are connecting are called as the stromal lamellae. Does this now ring a bell? What is a lamella or a lamina, lamella or lamina? It means a membrane. So inside the stroma you have these little tubes, membrane se bani hui tubes. So they are called as the stromal lamellae. They are basically flat membranous tubules. What they are connecting will become clear in the next 5 minutes. But these things which you see here, these membranous tubes, these membranous lamella, membranous tubes are called stroma lamellae. So now let's have a look inside the stroma and let's see what are these structures which you see here. So I'm just going to magnify this part now. And this looks like pouches which are stacked one on top of the other. So you can see here that there is this pouch like structure. Can you see this pouch like structure here? Can you see this? Now this pouch like structure, thulakos, do you remember that? So this pouch like structure is now called as thylakoid. So a thylakoid is an organized flattened membranous sac. So it's basically a chapta pouch, chapta theli. So this chapta pouch, flattened sac, thulakos is called thylakoid. And you can see that these pouches are stacked one on top of the other. So you will notice that all of these are thylakoids. And inside these thylakoids, you are going to have the pigment which is chlorophyll. So these are the ones which are going to contain chlorophyll, and that is why they appear green in color. You can see that all of them here are arranged in stacks like piles of coins. So you are taking one thylakoid which is like a pouch, and you can see that it resembles like a coin, and you place one on top of the other. So you get a whole stack of coins. And this, dear students, is called as granum. So here you can see that this is one stack of thylakoids. Here is another one which have been cut. That is also a granum. And here you have another one which is another granum. Granum is singular. Together they can be called as grana. So the plural term here is grana. So inside the stroma you have these pouches, thylakoid, coin-like structures, which are stacked one on top of the other to form this building-like structure, which is going to be called as granum, and many of them together are called grana. Is that clear? Okay. Now, you will notice that if I was to cut through the building or through the granum, each individual thylakoid will be cut, and you will see that inside there is a space. What will be the space inside this thylakoid called? What will be the space called? It will be called as the lumen. So what you see here, all of these spaces are called as the lumen. So just to have a quick recap here, you are going to have a plastid which contains mostly chlorophyll, which will be called as a chloroplast. Chloroplasts are usually found in the mesophyll cells of a leaf, can be anywhere from 20 to 40. In unicellular organisms like the green algae Chlamydomonas, you may only find one. They are going to be, a typical chloroplast is going to be anywhere from 5 to 10 microns in length and about 2 to 4 microns in width. It will have an outer membrane and an inner membrane which is less permeable with something called as the intermembrane space. Within the inner membrane, inside the inner membrane, whatever space is present is called as the stroma. The stroma will contain the various chemicals required to make glucose to do photosynthesis. The stroma is also going to contain the pigment chlorophyll. Where is the pigment chlorophyll? Inside pouches called as thylakoid, which are stacked one on top of the other to form a stack which is called as a granum, and many of them together are called grana. If you were to cut the thylakoid, the space inside is going to be called as the lumen. Are all these points very clear to everybody now? Okay, moving on. So now you can see that this structure here is going to be called as the granum, and these individual structures are called thylakoids. This is now going to become even more clear that what is the stroma lamella connecting. So the stroma lamella now you can see is connecting all these stack of coin-like structures, so it is connecting the grana. So the main function of the stroma lamella, the flat membranous tubule, is to connect the various grana together, as you can see here. As you can see here everywhere in this diagram. So finally, how does this look inside the electron microscope? So this, of course, what I have been explaining to you is an illustration. In the electron microscope, this is how it looks like. So I think now you can understand how the chloroplast looks in the electron microscope. These here are going to be the various grana made up of these individual coin-like structures which are thylakoid, and these connection that you see here are the stroma lamellae. The connection that you see here are the stroma lamellae. So inside a plant cell, where is the chlorophyll? The plant cell will have cytoplasm. The cytoplasm will have organelle called plastids. The plastid which contains more chlorophyll is called as chloroplast. The chloroplast will have an outer membrane and an inner membrane. Between them there will be an intermembrane space. Inside it, it will have the stroma. The stroma will be made up of many building like structures which are going to be called as grana. Singular will be granum. Each granum is made up of multiple thylakoids, and if you were to take the thylakoid, that is where you will have the chlorophyll within the thylakoid. The space inside the thylakoid is called as the lumen. So I hope that you can put all this knowledge and you can now apply it to this diagram that you see here on your screen, which is the electron microscope picture of the chloroplast. So once again, this is how the illustration looked, and this is how the electron microscope image appears to be. And with that, dear students, as far as all the cell organelles are concerned, out of these 8 cell organelles that we have to discuss, we have finished the first one, which is plastids. So I hope you have understood everything that we have done today, and I cannot wait to see your smiling faces in the next lecture. Thank you, dear children. Good day. God bless. Work hard. Be nice.